Okay, so thanks a lot for inviting me to this conference. So my talk will be really like a little bit connecting the talks of Rama, the first day, then of Joe, and of Joachim, and what Remy told us yesterday in the discussion. And it was to answer your question, Daniel, if we really use this machine. Yes, we really use them, and indeed they are very, very useful and nice. So why they are nice for us? Just because they are quite simple, so they can be inferred from data, but then you can do like math on them, like the statistical physics and spin glass math we are used to. And so my goal is to show you, as you can really apply them to two real cases. The first one is a small protein domain, actually the one which is called a WW domain, on which Rama has done very important experiments so that you can design this domain at the beginning. And then I will, if I have time, just to go to a more a larger protein, a more complicated case, which is the receptor binding domain of the spike protein. So our goal is to study and predict evolution in some sense from model learned from data. Okay. And I will focus on the idea of like mutational paths, so really similar to what Joachim has introduced. And this is a very nice example of a path here. So this is a very important work in the 1970 of John Maynard Smith, and introduced these ideas of paths, fitness landscape of protein as driving evolution, as a word game. So you start from a word, which is a word here, and you go to gene. At each step, you just change one letter. So you start by changing the DNA and so on. And you have to keep each of these words meaningful. So you have to go in high peaks in fitness landscape. So this is what we are trying to do with real protein. So this is a like picture of these paths we are designing. And so what we'll do in this, the first part of the talk, just we will anchor the starting and ending point into two real protein, and then we design, so we like invent the other proteins such that they have a high fitness. And so I've shown here three parts. We will, for example, three examples here. They go to different place in the sequence space, and this is one direct path in the sense that you change letters just which are just in the first or last protein, so you are not allowed to go to other letters. And then there are non-direct paths in which, like, for example, you have some letters which are different from the one at the beginning and the end. So we, with the Rosetta-Bosma machine, we can design these paths, and I will show you how. Okay, so this is the aim of this talk. So I would like to thank my collaborators, so Remy Monasson, Jorge Fernandez de Cosio Diaz, and our student Eugenio Mauri, Pierre Guillaume Bron, and also so I will show that on WW domain we have tested this path thanks to the collaborators, experimental collaborators, Marco Ribezzi and Ahmed Rehan at SPCI in Paris. And if I have time, I will go to SARS-CoV-2 protein, and my student Marianne Huot make all the works on SARS-CoV-2. Okay, so let's start. So proteins. So Ram already described it, but so you can see a protein just as a sequence of amino acids, which has a lot of constraint to work, so it has to satisfy all these constraints. First of all, it has to fold in a given structure, and then it has to have a function, for example, here to bind a ligand. So we are at the notion of ligand, it's just the peptide it binds to. Okay, so in the sequence, all these constraints are encrypted, so the stability, the affinity, in the sense that it has to bind to the target ligand, the specificity, it is to bind in a specific way to this ligand, particular ligand, and sometimes once it bound, it has to promote some reaction, so it can also have allostery, for example. So we want to understand how all these constraints are encoded in the sequences. So how do we do it? We start from all the sequences which are collected in the databases, for example, PFAM, on this particular protein. So this is called the multi-sequence alignment, because all the sequences are aligned, and so you will see a sequence as just a sequence of letters. So there are 20 possible letters, but there is also this symbol, which is a gap symbol. So just if you have a shorter sequence, you have to put a gap somewhere, just to say here there is no amino acid for this sequence. Okay, and all these sequences for us are these points, so they are all functional because they have been sampled on different organisms, living organisms, and we want to extract from these points our fitness landscape, which is just a function which is high on all the sampled points and is low on the other points that you don't have sampled. Okay, so this is just the probability that a sequence be a good sequence for this protein. So it's a member of this family. So this is our fitness landscape, which we infer from data. It depends only on the sequence. So the V are the amino acids at each position, N is just the length of the protein. Okay, so of course, Remy has shown how you can, so this is called unsupervised learning, in the sense that you have no label, you decide that all sequences you have are functional and are good points in this fitness landscape. So what we have worked a lot on this kind of inference, and once you have this fitness landscape, this is very convenient because you can do several things. For example, you can predict what happens if you change few amino acids on this on a given sequence, on a given wild type. So you predict the change in functionality, for example, when you do single or double mutation around a wild type. So this is really similar to what Joe was studying. And then also you can sample from this probability. And when you sample from this probability, you obtain like new sequences which have not been sampled through evolution. So you design new sequences. And also what we will do now, it's a more ambitious goal, is just design a transition path which go all around the functional space, but it goes from one given specificity, so the capacity of some of this sequence to bind a given ligand, to another specificity, which is the capacity to bind another ligand. So we want to, from the inferred landscape, design not only one protein, but all proteins which are able to make this transition. Okay? Yes.
Very simple question, I didn't get it. The prior knowledge from data, what should be that we could make a good suggestion for the landscape that made the data fit on it? Is there any prior knowledge that you should gather from data to know?
No, we just as like prior, we put some like regularization. So the number of data is important. So this make us regularize the model in such a way we don't overfit the data. But apart from this, we don't put any prior.
How we know this rugged landscape could be a good fit for this data?
We just then, so there is a very practical way of doing it, like for example, design a sequence and then see if it's functional or not. So this is the way we use. And what we do when we fit the model, we are sure that we are not overfitting. And then also we see what we want to just check that the like 1 point statistic, like the frequencies correlation, 3 points correlation of this multi-sequence alignment are well reproduced by the model when we do sampling. Okay. So for the moment, there is only statistics and the ability to reproduce our data. Okay, so we have worked a lot on Boltzmann machine in general, but how here I will just focus on the restricted Boltzmann machine. So Remy has introduced it yesterday, but basically they are very simple 2 layers networks. And the nice thing of this machine is that you go from the sequence space to one hidden units like space, which is like the feature space or the representation space. And normally this space can make you already a dimensional reduction of the sequence space. So normally the number of hidden units that you fit from your model, but just by cross-validation, is smaller than the number of the length of the protein, or more or less of the same order. And we'll see how you can, some particular hidden units represent really some feature the protein should have. Okay, so what you do practically, so you have this sequence Vi, then you project on the weights matrix. So the weights matrix is Wi mu, so it connects each amino acids on the sequence, so I to the hidden unit mu. And so when you project a sequence on the weights attached to an hidden unit, you compute the input to the hidden units, and then the units will activate or not, depending on the input it receives. So it will detect some pattern on your sequence.
So just a clarification on notation. This is linear, so I is W times matrix times mu?
Yeah, this is a matrix
linear,
which depends on I and mu. So it has 2 indices. It depends on the position. So these are exactly these vectors. So for one given mu, this is this vector. And then it's the matrices of all vectors attached to all hidden units. Okay?
It's linear.
It's a function in this case because these are Potts units. So it can take 20 value. So for each of these value, you have one the projection. Okay. For like binary units, it will be time. Okay, cool. So then, so the probability of having a given sequence, so this is the nice stuff about RBM. You can like write it like in two lines. So it's like unlike this deep model in which you cannot write. This is very simple writing. So the probability of a given sequence is thus the integral over possible value of the hidden units of these probabilities, which depends both on the sequence and on the hidden units. And it can be written as an exponential of an energy minus an energy, which is has local fields potential plus potential, which depends only on the input of each hidden units, which depends on the sequence. Okay, so you have really a nice way of writing this formally, this probability, and this potential is just the logarithm of the integral over hidden units of just these two terms. So this is the terms of input in hidden units, and this is the term of potential, which determines the way the hidden units activate. And as Remy has explained, there are two important facts. First, when this potential is quadratic, you find back the Hoffman models, where the number of patterns you store in this network is the number of hidden units. And in this case, when this is quadratic, the most likely value of H is depend linearly on I. So it is like linear hidden units. When you have more complicated hidden units, more complicated potential, you have like rectified hidden units, so which, for example, do not respond if the input is too low and then activate linearly. And we use just to parameterize this potential with like 4 parameters for each hidden units. So this is just to say that this machine seems very simple, but it's quite expressive thanks to this flexibility in adjusting the potential. So it's not Gaussian network. Okay, so this is the basic of the RBM. And so now what we do, we have learned the parameters of this RBM on this W domain. So this W domain, as I was saying before, was studied, for example, in 2005 by Rama, who showed that indeed you can generate a functional protein by simply by constraining the generation to reflecting the frequencies and the pairwise correlation in the multi-sequence alignment. So we started from this protein because a lot is known. Rama has already done some experiments to characterize, in particular, some sequences for their specificity. So the specificity is just that some of these, so this family break in like subfamilies which recognize different ligand. So it's very important in the functionality of this protein that there are all these different ability of recognize several ligands. For example, in human genome, there are a lot of these binding modules, which are very important for these properties of recognizing these different ligands. And so you can more or less cluster, identify different group of proteins according to the ligand they bind. So this is more or less the property of the ligand, okay, it has to be rich in proline, but it's not always the same amino acids. And these are like 4 groups which have been identified experimentally as binding to these different ligands. And we have some of annotated binding specificity, for example, thanks to work of Rama. And also, you can do like principal component-based analysis or the sector analysis made by Rama, and you identify this position here with a star as very key position for this binding specificity. So you know that some of these amino acids are important for the specificity. Okay, so what we have done is train on this multi-sequence alignment, our RBM. It was like 2,000 sequences. It's not a very, very enormous amount of sequences, but it's not small. And then what we have done, it's like identify 2 particular hidden units, which are the hidden units number 3 and hidden units number 4. And when you project the sequences on these 2 hidden units, you cluster them according to the specificity. So here are all the natural sequences, and the color correspond to the one which have been annotated, so which are known to be of type 1, 2, 3, or 4. You see that type 2 and 3 are not very well distinguishable, but type 4, there are very few annotated sequences. Type 1, there are a lot. And our RBM makes exactly this clustering like a PCA or something like this. It's a more sophisticated PCA. So the inputs, again, are just the projection of the sequence on this weight vector associated to hidden unit 3 and hidden units 4. Okay? So this is very nice because these 2 hidden units, by their activation, are able to detect the specificity of single sequences. So, for example, if the hidden units 3 have like a positive inputs and hidden units 4 have negative inputs, it will detect that this sequence is of type 4. Okay, yes?
But yesterday, Remy mentioned that depending on the number of hidden units, the representation can change vastly. So what is your rationale on how to choose the number?
So our rationale is just to like fit the data well and avoid overfit. So we do cross-validation. And in this case, so you know what is cross-validation. So we have a test set and a train set. So we test if our model works well on the test set. And here, in this protein, we had a number of hidden units of the order of the number of amino acids. And you see that we are in a regime in which you interpret the weights, in particular these 2 weights, nicely, as really what the amino acids which are important, the binding motif for the ligand. So we wanted to identify in these weights some, so have some interpretation of the weights. And so this was a good number of hidden units such that you can interpret well the weights. And I will show next slide indeed what I'm meaning by interpreting the weights. So the weights are quite sparse and they give us what are the important amino acids to be in the different binding specificity classes you have in your protein. So in particular, if, for example, you have an histidine in position 21, you will have a negative, if your sequence have a large overlap with this pattern, it will have a negative input on the hidden units tree, and so it will be of this type, of type 1. So this, so in this regime we choose, which is depending on the regularization, we can really interpret the weights as like binding motif. So you have to have these amino acids in this position to have like negative inputs to like hidden units 3, so to be of class 3, okay? So it's really nice because you can interpret the weights in terms of what are the important amino acids for the specificity. So these are the two. So this is exactly the same representation, another way of visually represent this vector, because it has like 20 possible value, because it's a Potts model, and each value is just the 8 of each letter, okay? So it's just a way to represent the weights to have which are the pattern. So it's exactly as in an Hopfield model. If you want to be a sequence of type 1, you have to have this pattern here in the sequence to be a good sequence of type 1. So it identify the minima of this fitness landscape, how they should be, okay? Exactly as the memory of your Hopfield model. And for each hidden units, you can have different interpretation. For example, some of them represent also contacts and things like this. Okay, so let's go to the mutational path. So first of all, so we can fix like one hidden units, the pair of hidden units to -3 and 1, just to generate protein with a given specificity. And then we, what we wanted to do was just to generate paths. So generate paths which go from the specificity, for example, of type 1 to the specificity to type 4. So which exactly as I have explaining at the beginning, have to have a good RBM likelihood, so a good score. Yes.
I think one of the things about that work that I was really impressed with was the possibility that you could factorize the sequence motif that gives you different classes of specificity, which is actually an old problem. You take a protein family, it's the same structure, basically, and it has the same binding mode and everything, but then it has to do many different functions. And so, you know, the famous example is that the human growth hormone receptor, which binds 2 different hormones, and the story is that they bind in the same place, but the pattern of forces that stabilize the 2 are actually completely decomposed and separate, right? So I was just wondering if that kind of like feature that you see there would give us a way to think about how we can even like encode dual functional proteins. I mean, is it possible to, do you think it'd be possible to create synthetic ones that are actually doing more than one function? And maybe those would be kind of the bridges between the different specificity group.
Yeah, sure, yeah.
I guess we never really tried to do those sort of experiments, right?
Yeah, yeah. So what we
just to clarify the question,
yeah.
By the factorizing, you mean that the costs and things of one part of the protein is sort of independent of the other part, so you can adjust one.
Yeah, yeah, exactly.
They don't work separately; they'll work together.
Yes.
Correct. Yeah, so you can adjust one without messing with the other.
So it's just that you factorize the specificity on a group of amino acids which are not nearby on the sequence, because you have to have this motif and also this one and so on.
I mean, it's not completely obvious because just from the first order sort of constraint like that, it's hard to say these are actually possible to do adjust independently. but it would be an interesting thing to try.
Right. So this is exactly why we wanted to do the path. You see something related. So we wanted to change specificity and see if in the middle you see like functionality which is like promiscuous, so you can bind both ligand of type 1 and type 4. And we'll see that indeed you can achieve this. So one important thing is that, as you can see here, the motif for like binding of type 4 is here and is in a different binding pocket from the motif of binding 1 to 3, as you know. Okay, so what we did, because time is passing, is just we design paths with the two conditions that RBM likelihood should be large for all the sequences in the path, so they are scored as good functional sequences, and you can mutate one or few amino acids along the path at the time, so you can not have big jump in your fitness landscape. So to do this, we just consider a free energy in which you have the path. So the T is the length of the path, which we fix, capital T. So for each small t step on the path, the RBM energy should be low, so as a good sequence for the RBM, and also the elastic cost of going from one sequence to the next one should be small. So this elastic cost depends only on the overlap between the sequence at time t and the sequence at time t minus 1. So it's called the overlap, you know why. And so it's just the fraction of unmutated residues between two consecutive sequences along the path.
So why is that you're just calling it an elastic cost? It's not actually.
Yeah.
Can I ask, why do you do this instead of just freezing positions and then mutating in the RBM itself?
I have to mutate, but with the condition that you don't want to mutate too much.
Freezing the positions and conditionally sample, right? The nice thing about the RBM is you can freeze and conditionally sample the rest.
Yeah, what we do is just exactly what we do. So we start by proposing a path, then we propose a mutation, so a different sequence along the path, and we accept or not according to Monte Carlo sampling. So this is a quite efficient way of sampling all the path. And it can end up to going through different portion of the sequence space. So it's not a direct path, it can be also non-direct path. Okay? So, just to show you, so this was an interest. So we designed several paths from type 1 to type 1, type 1, type 2, 3, type 1 to type 4, and I will show you this part because it was interesting, because it goes through a region of sequence space in which you don't have any natural sequences. So we were wondering why this was this way, and if these designed sequences were really functional or not. So we have a first tried with like AlphaFold and with protein MPPN to see if they were, how they were scored. And they were good sequences, yes. And yes, in the middle, just they were indeed promiscuous in the sense that they were able to bind according to protein MPN the two different ligands. Yes.
So if you look at the barrier in energy along this path, is of which order in N in the size of the sequence?
So it's order 1. So you.
Okay, so these are small barrier.
Yes,
Okay.
yes. You do in such a way that each step is a good, so you have a good sequence. Okay. So this was very interesting to see if, so what we have designed a strange sequence in the middle of the path, which can bind the two peptides. So these are called promiscuous sequences and can be related to ancestral and not specialized sequences. So they are quite interesting from both an evolution point of view and just from a technological point of view. So design such kind of sequence. Okay, so what we did, okay, first this was for Joe, but he's not here. You can also reconstruct the phylogenetic tree in the space of every sequence. So this is by ancestral sequence reconstruction. So it's exactly what Joe explained. And you find several paths, and there is one path which is quite similar to the one which we have designed. And you can see that indeed the internal nodes along the path are not too far from what we have designed. So this tell you that it can be indeed this kind of ancestral sequences. And then we have
roughly the total number of mutations along that fellow.
So what we would design, the total number of mutation, it was of the order of like, you see, we test 22 mutation. It was of the order of the number of nucleotide in the sequence, or the length of the sequence. So basically, you mutate at each step one amino acid.
No, but if you look at the phylogeny,
comparing
with how many mutations are there along the phylogenetic branches?
In the phylogenetic branch, so here you see it's of order of some tens. They can be longer, like 30, 40 or something like this. But up to like 50. So they are these dots. Okay, so then just, I will not go to the SARS-CoV-2 stuff, but the test of the designed paths. So this was done in SPC by Marco Ribezzi, Ahmed Rehan. So you can just probe the binding by fluorescent in a tube. So you have all your sequences, and then the given ligand, and if they bind, they are detected by fluorescence. If they not, they do not fluoresce. And so it tested the specificity for 3 ligands, one of type 1, 2, and 4, so the response of the protein. And so this was, for example, first path, which had to go to one wild type to another wild type on the same specificity class. So you see that these 2 points here and here are the real protein, and they have a score RBM, which is smaller than some protein along the path. So in the RBM, if you decrease the sampling temperature, you can even design proteins which are more stable than the natural one. And then you can also give a score which is given by an RBM trained only on class 1 protein. So this will be the specificity score in our case. And these are the experiments. So all the sequences we have tested, so they are the one in red, respond and fluoresce. And so they are all functional. So to test that this was a real good path, what we have done is scramble the path. So scramble the order in which the mutations are done. And if you scramble the path, you don't have any functional sequences. So it's really important to keep the epistasis to do the mutation in a given order to have functional sequences. And so then, so this is the test.
Understand the data. So the claim is that What do you mean by scramble? Sorry.
The mutational path.
Yeah.
So we did in a path, so you start by a sequence, and then you do a mutation of amino acid number 3, then 4, and so on, for example. Scramble means that you start from the sequence, and then you first mutate the amino acid 4, and then the amino acid
The first
3.
and the last are the same, so they work.
Yes.
Regardless.
Yes.
Okay, I understand.
But if you the background on which
Manifold of.
so if the background in which you are doing the mutation is not the right one, you have not prepared the background with a given number of mutation, you will not have.
The RBM also says those are.
Yes. The RBM also says that, and these are exactly the shuffled path. In this case, only the 2, 1, and 21, the anchoring point are functional, or the other are non-functional. And this is an example of this path from 1 to 4. And you see that in the middle, this sequence 21 is both responding to peptide 1 and to peptide 4. And actually, some sequences here respond both to peptide 1 and 2. So what we have seen that
They all fold.
We don't know, because they just test the binding response, but if they don't fold, they will not bind. So. And according to the RBM score is like a good proxy for the stability. Okay, so the all tested sequences were functional, and they shuffled.
If you shuffle, you're saying all possible shuffles
Yes.
are not functional along the way, or is that?
Yeah, the shuffle, so if you have the one which correspond to our path or another path, they will be functional, but all what we have tested, they were not functional. Okay, because you don't have the right background to do the mutation.
But shuffling just means that you permute the order in which.
Yes, you change the order in which mutation happens, so you keep this number of mutation and change. Of course, we have tested the limited number of shuffled paths, so the sequence were the same number as the one we tested for our.
If you only exchange like 2 steps, would it still make it non-functional?
Just 2 steps. We have tried some specific mutation, and yes, which were just 1 amino acids away, and they were not functional. But they were chosen to be like in a critical part. Okay, but in general, sometimes if you do 2 like neutral mutation, you expect that they will be functional. Okay, so nice thing we can do with it, but I will not go into this just in one slide before, because we have the time is running out, that you can do mean-field theory of this like path. So just by a free energy of the path, which depend only on the magnetization, which are the input on the different hidden units of a given sequence. So you can use statistical physics to compute like this like mean field path, which depend only on the magnetization, which are the inputs on the different hidden units. And you see that indeed they go on the same region of the space. And this is nice because you can compute the entropy of path, so the number of possible functional paths. So I will end because I think time is 2 minutes. Okay, so just I can explain you this RBM free energy. It's very simple. So this is Legendre transform of the RBM potential, which so depends only of the magnetization. This is the elastic free energy, and then you have the logarithm of the partition function for each site, which you can compute because it depends only on the site, so you can compute by like transfer matrix, and this allows you to compute the entropy of the paths. Okay, so let's go to the conclusion. So we have done this in a much more complicated protein, which is the receptor binding domain of SARS-CoV-2 protein, and we have computed the escaping path to the antibodies. And I will not go through this, but just to say you that this RBM can be applied also to just more longer protein and with many different functions which can like evolve with time. So we want to, and we have also tested this protein with experiments. Okay, so, and we have used a lot the mean field theory in this case. So the conclusion, so I have shown you this restricted Boltzmann machine, which is an unsupervised simple architecture to infer model from sequence data. It is generative and it is interpretable if you choose well the number of hidden units, because the weights can identify important sequence motif, for example, for the binding specificity. And it's nice because it's the right way of going from the sequence to feature space. So for more complicated protein, for example, longer one, to find which hidden units is attached to a given functionality, you have to train your RBM to disentangle the weights to these hidden units with the other to be really sure that these hidden units, for example, will be the one for the specificity. And this we have a paper on this. So we have seen this transition path algorithm. And so this is the paper in which they have tested the transition path in the WW. So the paper in collaboration with the group of SPCI. And so this is a paper in which we have shown this path to escape antibodies for the receptor binding domain in SARS-CoV-2. So thanks a lot.
Yes.
How is defined this escape path, the SARS-CoV case?
Yes, so in the SARS-CoV-2, we didn't, so these are the escape path in the SARS-CoV-2. So we didn't post the final sequence. We imposed just the first one and the fact that you have to increase resistance against antibody. And then so you have a term that you add, which is the binding to the antibodies. And you can fit on some other experiments in which they have tested all this binding. So basically you have to add these terms in the probability that the sequence is good. So it's viable, but also bind antibodies. So escape antibodies, sorry. And then you design paths which increase this overall fitness. And you can see that this is like mean field theory. So these always are the path projected on the like two coordinates, and gives you variants which most like variants which escape to the newest, so the newest variants in the pandemic. So you go through these new variants.
Are you worried about the phylogenetic signal in the alignment of the RBD?
So the phylogenetic signal, we have not checked this perfectly. We would like to do, but in some cases we have go into some specific hidden units if you disentangle well. So you can also see some hidden units which gives you like the phylogenetic signal. So what kind of protein it is. So cluster the proteins according to phylogeny.
But in this case, it's presumably very dominant if the data that you trained on are from a short evolution. Like, is it a good intuition?
In the case of SARS-CoV-2,
Yes.
in the case of SARS-CoV-2, indeed. So what we did, we train on the pre-pandemic data, which are much diverged. So this helps you in like taking more distant like sequences at the beginning, and they avoid all this. So we use just pandemic just to check our prediction pandemic data, but we train on pre-pandemic data.
I know that like Michael Desai's group measured kind of a local neighborhood of one of these, and I'm struggling to connect this, but is there a way to validate?
Some yes. Or? Yeah. So so I know very well this work. So we have tested all possible mutation between two variants, and they have tested the binding specificity. And we have, I think, have we connected to this? I don't think so, but yes, it could be an interesting thing.
But these experiments, they're all possible mutations in the viral sequence, but then how broad a set of antibodies that are being tested?
Yeah, so there are a lot of antibodies. So in the model, we put like 30 antibodies.
I mean, in the experiments, how many antibodies?
So the group of Jesse Bloom has tested like 30 antibodies. So how each variant of the receptor binding domain bind or not on the antibody, and from this we have fitted this binding constant.
The 30 natural antibodies that are found.
Yes, but they are of the different classes, so they reflect well, like for example, the evolution of the pandemic. So when they arrive, so the classes are where they bind on the protein. So we fit the probability of the escape from exactly this data, and we fit the probability to escape to each of possible tested antibodies.
But where they're binding on the spike, which parts?
Yes.
Any other question? No? Okay, so let's thank Simona again.